Hello students, welcome to lab number 10. We're going to be talking about photosynthesis. This is basically the lab that we talk about the chromatography strips, we talk about the photosynthesis and cellular respiration within the leaf discs, photosynthesis experiment. That one you may have to watch online through this YouTube thing that I found. So let's go ahead and talk about the introduction. Photosynthesis is the process where plants take in sunlight, water, and carbon dioxide and transform that into chemical energy. So that's glucose, and byproduct is oxygen. That oxygen can be translated into the mitochondria. Some of that oxygen is transpired out into the environment for us to breathe in as well. So anyways, photosynthesis takes place in chloroplasts. Here membranous thylakoids are stacked in grana, surrounded by stroma. So the stroma is the liquid inside the chloroplasts, inside the second membrane of the chloroplasts that are surrounding the grana. During light reactions, pigments within the membranes of the thylakoid, like chlorophyll, are absorbing light energy. Water is split by that light energy. Oxygen is released when the water is split. Energy is captured in the light reactions. Going to the Calvin cycle, ATP is reduced. ATP is released into the cells, reduced NADPH, and then Calvin cycle takes that NADPH, and then ATP is produced there as well. Calvin cycle reactions occur within the stroma, in those membranes. During these reactions, carbon dioxide is reduced as well. Those carbon fixation reactions occur, and then those are fixed into those glucose molecules, and then those glucose molecules are stored into carbohydrates, into starches or oils or waxes or whatever. So here we have a chloroplast molecule, or chloroplast organelle. We have the thylakoids in those individual discs, right? All those stacks of thylakoids are called granum, right? If you have multiple granum, they're called grana. Inside the inner membrane there, all of the liquid inside that are called the stroma. We have the outer membrane, and then we have small circles of DNA. All those little circles of DNA are what we think of when they came up with the idea of endosymbiosis theory. So the circular DNA is what you can find in bacteria, and so in chloroplasts they also have circular DNA. So what they thought of was chloroplasts have circular DNA, bacteria have circular DNA, so chloroplasts may have come from an earlier form of bacterial colonies. That's something that you can look up later, or we can talk about later in the class. So the first experiment that is in your book, is in your manual, is the chromatography experiment. This is a technique used to separate pigment molecules. So in chlorophyll, in the chromoplasts, there's these different pigments, right? The chloroplasts don't just use one pigment. Okay, so they use chlorophyll A, chlorophyll B, xanthophylls, and chromophil, or carotenoids, right? So carotenoids are orange. They also use anthocyanins, but you won't be able to find it in this chromatography solution because anthocyanins are water soluble. The rest of these are non-polar substances. So anthocyanins are polar substances. The rest of these are non-polar substances. So chromatography solution is ether acetone, which is a non-polar substance. It's an oily type of substance. A leaf is rubbed on the strip of chromatography paper, placed in a test tube containing that ether acetone or chromatography solution. By the way, this solution is very volatile. It'll evaporate very quickly. So if we did this in the lab, we would probably do it in a vented station, or we'd probably do it with a plug on top so it wouldn't vent in your face. Anyways, so it's wicked up through the paper strip. When it encounters the leaf rub, it contains the pigment molecules. As the solution continues up the strip, the pigments will be pulled along with it. So the pigments that are pulled up through the chromatography strip that are the most soluble in this solution are also the most nonpolar. The pigments that are polar, that were not able to dissolve in the solution, therefore these remained at the bottom. So the most polar remained at the bottom, the most nonpolar got pulled up to the top. Okay, so beta carotene are the most nonpolar because it contained the most nonpolar molecular structures. Chlorophyll B and chlorophyll A are the most polar because it contains the most polar substances in it. So whenever it pulls the substance up, whenever that chromatography solution pulls the substance up through that paper, it does so as a molecular, kind of a molecular filter. So the beta carotene is the most nonpolar, and the chlorophyll B is the most polar. RF value is used to quantitatively compare paper chromatography results. An RFV, or RF value, is the distance traveled by the pigment over the distance traveled by the solvent. And in your assignment paper, I give you all of the numbers that we have in this chromatography strip. Okay, and you should be able to find all the RF values there. Well-dissolved, fast-moving pigments will have an RF value close to 1. Nothing will have an RF value more than 1 because no pigment will move faster than the solvent that's run. Next, we're going to go to the next experiment. So this is the disc experiment. Photosynthesis occurs in the presence of sunlight or light and oxygen. However, in the absence of light, photosynthesis will not occur and oxygen will not be produced. Right? It takes light to split apart that H, those two H and that O for that water and oxygen. Right? Cellular respiration uses oxygen as a reactant, though. So cellular respiration occurs when light is present or not. We can use an experiment to show the effects of photosynthesis and cellular respiration. So this is the leaf disc experiment. In the leaf disc experiment, you punch holes in the leaf, you remove oxygen from the leaves, you place it in this tube, in this syringe. Basically, removing oxygen from the leaf, you just basically inundate the leaves with water, and then you place the leaves in this bicarbonate solution, and then you place them in light. Usually they're going to sink. Okay, why would they sink? Because they don't have any oxygen in them. Well, when they're in the light, they're going to start to rise, right? So why would they start to rise? Because now they're starting to get oxygen into the leaves. They're starting to get oxygen trapped inside those leaf discs. Okay, so now that you kind of understand that, you can go through this experiment. You can go through the experiment through YouTube. This is Bozeman Science. And you can see how that works on the photosynthesis lab. All right. So the cellular respiration experiment. So now that they're all floating, you can put them in the dark. So now they're all floating, you put them in the dark, and you leave them there overnight. What do you think that's going to happen there overnight? Well, the oxygen is going to be used up, right? They still have mitochondria. So what cell organ is involved in cellular respiration? That's going to be the mitochondria. What gas is being used up? That's going to be the oxygen. And then is ATP being used aerobically or anaerobically? That's aerobically because it's using up the oxygen. If oxygen is there, it's aerobic. Go ahead and go through the questions in the lab assignment that I posted. Here's some more review questions for Lab Practical 2. Make sure that you go through those. Your next Lab Practical is going to be on the 28th, so make sure that you study for that. Also, common assignment is due on the 26th. Make sure that you do that. That is individual. So make sure that you have that done and turned in to me by that day. All right, if you have any more questions, please let me know, and I will see you later.